It's now my pleasure to introduce Dr. John Clark, who's the Co-Director for Science and Conservation at the National Tropical Botanical Garden and soon to be Executive Director of the Center for Plant Conservation. John.
Hi, everyone. Thanks again, Chipper, for that introduction. Again, my name is John Clark. I'm Co-Director of Science and Conservation at National Tropical Botanic Garden, for today at least. As Chipper mentioned, tomorrow I assume the leadership of the Center for Plant Conservation with Dr. Peter Raven as the chair of our board, and I look forward to an exciting future, not only working under the auspices of the CPC, but also working intimately with the partnering institutions around the country, including the National Tropical Botanic Garden, to continue the valiant work of plant conservation in and among those gardens around the country. Chipper likes to point out that he's on the chair of the board of trustees for the Center for Plant Conservation, so in effect he's still my boss even after today. So today I have the honor to introduce a couple of my mentors and colleagues who have been working in plant conservation and plant research for many years now and have been able to effect wonderful change by working not strictly in academia, but actually bridging out into the world of botanic gardens. Botanic gardens that as has been discussed, has been a real force in plant conservation as well as in basic research. Not only getting out there and doing the work that needs to be done, but also in training future scientists and future conservationists to do that research. I, fortunately, am one of those people that is a product of this system. And what I'd like to do is explain a little bit of my background and how I became involved in plant conservation as well as with the National Tropical Botanic Garden. I began my research on a group of plants that are found throughout the Pacific in the genus Sertandra. It's the African violet family. The family is known as the Gesneriaceae. This genus has about 700 species, and they're distributed from the Indian Ocean, the Nicobar Islands in the Indian Ocean, throughout Southeast Asia, and out into the remote islands of the Pacific. There are about 700 species across this broad range, but any of those species in particular are very narrow endemic species, often restricted to archipelagos, more often an island, and sometimes even a single valley within those islands. So species, even though the genus is broadly distributed across about a third of the circumference of the planet, the species themselves are under high risk of extinction because of potential changes happening on a local scale. For that reason, it becomes a wonderful model to understand patterns in diversification in plants, as well as look at ways that we can start to work to better conserve those species. I began my dissertation research in 2003, and shortly thereafter I made my first trip to Hawaii, where I got to go out in the field and see these plants in their native habitat. I was on the island of Kauai, and I met up with National Tropical Botanic Garden scientist Steve Perlman, who Chipper had just mentioned, as well as Natalia Tangolin, at that time a new field botanist for National Tropical Botanic Garden. They took me, along with my advisor, Eric Rolson, into the hills in North Kauai, and I found my first populations of Sertandra growing in the Limahuli Garden Preserve. They were wonderful plants to see, and I became very excited about this and passionate about moving forward with my research. And over the course of the next 5 years, I started traveling around the Pacific, including to places like Samoa, the Solomon Islands, Fiji, to collect these species and to learn more about their relationships, how they made it out into the Pacific, and to learn what I could do to help ensure their survival. In working on these projects, I would often do basic research, historical research, looking at where those researchers, those field botanists that came before me went to collect these species. Botanists, as we go on the field, continue to do this kind of work where we look who has looked where for these plants, and then go and try to recollect those species and also document additional populations throughout their range. Alarmingly, what I found when I would go to these places is a large number of these localities where these species were once found no longer existed. The place, of course, was there, but the habitat had been altered in such a way that the species that I was looking for could not exist there. This would happen for a number of reasons, particularly from deforestation, but also from the threats of invasive species. This is obviously an alarming trend and not unique to the group of plants that I work on, but a factor that we're seeing affecting diversity across this planet. As I continued to do this work and collected more and more species where we did find them, I built up what is known as a phylogeny, the relationship of species within this genus, and also worked on the biogeographic patterns, using this phylogenetic information, how those plants are related to one another, to build an idea of how, when, and where they disperse through the Pacific. This information was wonderful to figure out for that group of plants, but it also gave us information on understanding dispersal patterns and evolutionary patterns in plants across the Pacific. That in and of itself was excellent work, but as I continued to do this, I would find more and more that the concerns of what are happening to these species is something that was overwhelming me more and more. When I was trying to piece together this project, initially I was very excited about the opportunity of just being able to go out in the Pacific and venture to all of these islands and have these wonderful trips. But more and more I became more interested in being able to collect those data. As an evolutionary biologist and a systematist, I am basically building a puzzle with pieces that are available to me. Imagine starting on a really large puzzle and a number of those pieces were already missing before you got started. Imagine further that if you're working on that puzzle with all those missing pieces and someone's over your shoulder grabbing pieces and chucking them out the window. That's kind of what it's like trying to build an evolutionary hypothesis of species in today's world where we're seeing alarming rates of species loss across the planet. For those reasons, I became ever interested in conservation principles. And when I was invited to come on board the National Tropical Botanic Garden in 2013 as a staff scientist, I wanted to look not only at biogeography, not only at systematics, but see what I could do to more effectively make changes for our planet through science and conservation research to ensure the survival of these species. Working with Chipper Wickman and Dave Lawrence, as well as Warren Wagner and the amazing staff at National Tropical Botanic Garden, we worked to actually improve our conservation programs by merging our science and, at that time, independent conservation programs, and also looking at more creative ways that we could explore both scientifically and measurably how we can actually affect real conservation outcomes. In this way, the National Tropical Botanic Garden has been a real leader and continues to be a leader as we move forward in this very uncertain 21st century. Today, I actually have the honor of introducing a couple of my mentors, Dr. Kay Havens, who's Director of Science at Chicago Botanic Garden, and Dr. Lucinda Mcdade, who's the Executive Director of the Rancho Santa Ana Botanic Garden, as well as Director of their research programs there. These two wonderful women scientists have been influential to me in my personal evolution as a scientist and ultimately a conservation biologist. Their expertise and their ability to not only innovate scientifically, as well as bring in new recruits and train the future scientists and conservationists in our fields has been exemplary. Their programs that they've developed at Chicago and at Rancho Santa Ana Botanic Garden are excellent examples of how small to medium-sized botanic gardens and relatively new botanic gardens are able to affect real and lasting changes in our understanding of the natural world, and also to develop ways that we can implement conservation practices in this country and around the world. So with that, I'm going to introduce Dr. Kay Havens. She will speak first on current practices in plant conservation in botanic gardens and talk a little bit about her research programs and her vision for plant conservation. And then seamlessly, Dr. Lucinda Mcdade will talk about her work at Rancho Santa Ana Botanic Garden, as well as her vision for the future of plant conservation and how botanic gardens will help meet that need. Thank you.
Thank you, John and Chipper. It is indeed an honor to be standing here today with so many of my favorite botanists and botanical garden professionals. I was at a meeting a couple of weeks ago, and Steve Blackmore said, "Botany is the foundational science for a sustainable future." And I thought, what a wonderful way to say what we all know, that all of the environmental grand challenges that we face require botanical knowledge and their solutions for everything from climate change to food security require effectively conserving plants and plant communities. The global strategy for plant conservation provides an amazing roadmap for us. The GSPC is a program of the United Nations Convention on Biological Diversity, and has 16 action-oriented targets that hopefully we are to meet by 2020. This week in Korea, the midterm report came out and said we're not on track and we're not going to meet those targets. But I'd like to think that this is a call for further investment, that we could meet those targets if we work harder and more effectively. The targets range from a complete online flora of all known plants to conserving 75% of our threatened species in situ and ex situ. And target 8, the ex situ target, is one where most botanic gardens are active. We know that plants are threatened for a variety of reasons: habitat loss, invasive species, overharvest, pollution, climate change. And these species are best protected in situ where they live, but ex situ conservation, or off-site conservation done by banking seeds, offers both an insurance policy against extinction and propagules for reintroduction and restoration. It's a low-cost method to preserve big numbers of individuals, and in situ and ex situ are not alternatives, but are complementary strategies in integrated conservation. In this time of global change, seed banking, I think, is one of the most rational things we can do. Seeds are truly remarkable. If you think about them, it's a little baby plant and its lunchbox all packaged up, ready to go, ready to sleep, designed by evolution to stay dormant for some period of time. And we can take advantage of this dormancy by drying and freezing these seeds for long periods of time. About 80% of our plant species are orthodox, which means they have seeds that can withstand this treatment. The remainder are recalcitrant, they can't withstand the desiccation, or intermediate, they don't live long in storage. But for the vast majority of plants, we can bank seeds. Most species with recalcitrant seeds come from the tropics. But there are a few, like oaks and chestnuts, from temperate regions. And for decades, I think the perception in tropical regions has been you can't bank the flora. And happily, we're finding that this maybe isn't the case. A recent study of the Hawaiian flora showed that only 5% of the species were truly recalcitrant. And this calls for much greater investment in ex situ capacity in tropical areas. In addition, advances in cryopreservation, including dissecting and freezing plant embryonic axes, are opening up long-term storage options for the remaining species. And we're at a point in time when virtually all species can be conserved off-site and for long-term if the time and effort is spent to do so. In the US, the nearly 40 gardens that make up the Center for Plant Conservation are actively collecting and banking our rarest seeds. And thanks to them, we are making good progress on our target 8 of reaching 75% of our flora in storage. Depending on how we define threatened species, if we're looking solely at listed species, we're at about 70% of those. If we define it more broadly to include all the nature-served G1 to G3 species, we're only at about 40%. And I think that shows us that we have work left to do. Beyond threatened species, many botanic gardens, including ourselves and Rancho Santa Ana, partner with Seeds of Success to bank the more common species, especially those important for restoration. Having adequate stocks of these restoration workhorse species banked from every ecoregion is an essential first step in improving restoration outcomes. And having this resource allows us some time for restoration science to catch up with restoration needs. The costs for doing this are feasible. We can do this. We estimate that it's about $250 million to bank the entire US flora. That's multiple samples per species and the associated research to grow those taxa. And while that sounds like a tremendous amount of money, and indeed it is, compared to some other things, not so much. It's about 3 hours of the Pentagon's budget. It's about 1/6 the cost of the Bellagio casino, and about 3 times the cost of Oprah's recent real estate purchase. So I was telling a friend about this the other day, and he said, I can see the headline now. Botany wins, the Pentagon becomes square. But we don't need that kind of investment. We just need a little sliver of the pie, even a crumb, and we can do miracles. We can do this if we choose to, and we need to choose to. Ex situ conservation by itself is not enough. The ultimate goal is to get this material reestablished in the wild. We're talking about seed banks with withdrawals and deposits and not seed morgues. An excellent success story is provided by the New England Wildflower Society, whose reintroduction of Potentilla robinsiana in the White Mountains was significantly responsible for its delisting. Given the threats natural communities are facing, we know that restoration is going to become even more critical in the next decades. And recognizing this, the Botanic Garden community formed the Ecological Restoration Alliance in 2011, and that's coordinated by Botanic Gardens Conservation International. Many gardens around the world are participating, including National Tropical, Missouri, Kew, Kings Park, and ourselves. And Botanic Gardens have several core competencies, including knowledge of the local flora, horticultural expertise, ecological expertise, and seed banks that make us ideal partners in restoration. The Ecological Restoration Alliance has the goal of completing 100 significant projects on 6 continents in the next 20 years. It's important to remember that one size does not fit all when it comes to restoration, that patterns of local adaptation and consequently seed transfer zones vary from place to place and vary from species to species. Practices that work in the tropics may not work in arid regions and vice versa. I think one of the most important things that will come out of the Ecological Restoration Alliance's activities is a better understanding of the commonalities and the differences in best practices for restoration around the world. We can learn a lot from restorations that have been in the ground for some time, and in our region, the 2 oldest prairie restorations were done by Botanic Gardens, including Curtis Prairie at University of Wisconsin Arboretum and the Schulenburg Prairie at Morton Arboretum. One universal question we're all facing in restoration is, should we change our seed sourcing practices as the climate changes? Should we source from a wider area? Should we try to predict what the climate's going to be like here in 50 years and collect seed from areas that match that climate now? Or should we continue to use local seed, which has been the gold standard in restoration for many decades? Making appropriate seed sourcing decisions will depend on many factors, including the species we're attempting to restore, the landscape context, the size and the urgency of the project. Our modeling indicates that in the eastern and midwestern U.S., obtaining appropriate materials will be a lot easier because our projects tend to be small, our seed zones tend to be large, and our plants tend to be predictable in that they're going to move, most of them, north and east as the climate changes. The situation in the western U.S. is a lot more complex, where the scale of restoration projects is immense, hundreds of thousands of acres sometimes. The patterns of local adaptation are more fine-grained, seed zones are smaller, and the way plants move under climate change is completely unpredictable based on our modeling. I think Botanic Gardens have a really important role to play in testing assumptions for seed sourcing under climate change. And I'm happy to say that we've partnered with Missouri Botanical Garden and Minnesota Landscape Arboretum to create a latitudinal transect of common gardens where we'll test several native species seed germination tolerances in those different climate zones. The Arboretum at Flagstaff has created a similar transect of gardens along an elevational gradient known as SEGA. And Missouri Botanical Garden has proposed that Botanic Gardens serve as test sites for assisted migration. A recent study in Europe tells us that in reality we're already doing this, that 73% of the native European taxa are being grown on average 1,000 kilometers north of their northernmost range limit in nature, in botanic gardens and in commercial nurseries. I think we're facing a paradigm shift in restoration right now, where we're no longer trying to go back in time to restore something that once was, but we're looking forward. And I wonder if creating future-proof plant communities isn't really more pre-storing than restoring. But regardless of what we call it, the challenge of developing and maintaining resilient and adaptable plant communities is ever more challenging and ever more important in our changing world. I'd like to leave you with one final thought. Plants are not optional. We know that, right? All life depends on plants. We now have the scientific capability to save any plant species, but what we lack is the political will and the funding to save them all. If we, the people in this room who care the most about plants, don't make our voices heard, nothing is going to change. But we're the agents of change. So we need to do this, and I look forward to working with you to make those changes. Thanks.
Hi, everyone. I'm Lucinda Mcdade. I'm the Executive Director of Rancho Santa Ana Botanic Garden in Southern California. We are the largest garden dedicated to the remarkable flora of the state of California, about 1/3 of the taxa in all of North America. So we have a major mission. And as Kay very nicely alluded to, in the West, patterns of plant distributions are quite complicated, and it's very challenging to predict what's going to happen tomorrow, much less more distantly in the future. Our garden is a display garden. We display the flora of California, but we are also a major research institution and a collections institution, having a herbarium as well as a major seed bank, library, and laboratories. I have the perhaps enviable task of talking about my vision for the future of botanic gardens in our complicated and ever-changing world. And I want to start by saying that my vision is simply that all botanic gardens should accept the challenge of being agents of change. All botanic gardens have roles to play, as remarkably different as botanic gardens are, sort of spanning the spectrum from pure pleasure gardens to much more complicated ones that have a lot of entities that involve research, that actively embrace roles in research, conservation, etc. I think that botanic gardens especially have a very, very important role to play, a unique role to play in overcoming green blindness, which is another way of phrasing the problem that Kay just alluded to, of many people simply not getting plants. Even people who love animals have a hard time sometimes understanding that plants aren't just the green background, the fuzzy stuff that the animals perch on or stand against or what have you, that plants are absolutely essential for the people of this planet and for the well-being of the planet. We simply have to overcome green blindness, among other things, very practically speaking, if we're going to get our tiny sliver of the pie, our fifth of the pentagon that we need in order to accomplish great things. So battling green blindness is essential, and every single botanic garden, I firmly believe, has a major role to play in this. We've got to achieve this. I'm not going to say a huge amount in my brief remarks here today about collaborations, consortia, networks, and that sort of thing. They are vital, and it should be understood as implicit in everything I'm saying that no one botanic garden does this alone. Again, Kay alluded eloquently to a number of very important consortia and networks that are forming. These are vital. They need to be formed. They need to be flexible. They need to be very effective in getting things done. So, some specifics for how I suggest that botanic gardens think about the future. I think we need to start by embracing all land plants. I would love to see a pleasure garden of bryophytes or lycopods or ferns. Ferns are common in botanic gardens, but really we are not doing a very good job, I would argue, at preserving all of the lineages of land plants, all of the remarkable diversity of land plants that share planet Earth with us. I think we need to take responsibility for plants that are challenging to grow. Kay mentioned one aspect of that, the plants with recalcitrant seeds. And we also think immediately of tissue culture, but I would like to see us also think more actively about plants that are parasites or hemiparasites, plants that are obligate mutualists, trying to more effectively bring these things into ex situ conservation. I love to ask people why you can't buy a seed packet of Indian paintbrush. As beautiful as Indian paintbrush is, of course, it's a hemiparasite, and so that doesn't work. They're challenging to grow, but it can be done. Clearly, we need to continue the embrace of modern methods as well as optimizing traditional ones in order to fully achieve our missions in conservation and research. We can enhance the conservation value of our work by embracing these methods. So, for example, the more that we know about our ex situ collections, limited as they usually are in terms of genetic samples of populations, the better off we are in terms of understanding their value and comparing the ex situ collections in one garden to those of another. I think we need to begin thinking about planned breeding programs of plants that are few in collections but maybe represent different genetic stock. Here the zoos are way ahead of us. We all know they move the pandas around to mate with each other and that sort of thing. And we need to, I think, do the same thing with plants. And these will take advantage of our what are now standard DNA techniques, but will also very rapidly take advantage of next generation sequencing, which is just about to become quite inexpensive for the acquisition of massive amounts of data. Again, no one garden is necessarily going to do all of this alone, but through collaborations and networking, this can get done. We can also, many gardens that have conservation programs off-site also are involved in situ studies. My garden recently undertook a study of a Hazardia, a composite of Asteraceae, in Southern California, of which there's only a single population in all of the United States. That's natural, but there were 4 outplantings done by some well-meaning people some years ago, but without any record having been made of where they got the plants from, whether even it was seeds or cuttings or whatever it was. And so, for all anyone knew, it was seeds from one mother plant in these now hundreds of outplantings. And we were able to show that using fragment analysis, DNA fragment analysis, that it's basically a subset, the outplantings were basically a subset of this one source population, and thus do have somewhat limited but still some conservation value. So we can assess the genetic viability of natural populations in this sort of way. Again, this kind of work is about to get much, much more efficient and powerful going forward with the methods of next generation sequencing. We can also use big data approaches and new methods, modern methods that are becoming increasingly available and increasingly efficient to enhance our research, those gardens that do research to increase our knowledge of plants worldwide. So, for example, we are increasingly contributing as individual gardens and as networks to very large online available data sets of vouchered plant specimens. The Consortium of California Herbaria is nearing 2 million specimen records for plants in California, available online instantaneously, and about 2/3 of them now are mappable, have latitude and longitude associated with them, and we're rapidly populating the full data set with georeferencing. This gives you an incredibly powerful tool to ask things like where specimens are not from, which lets you then say to a graduate student doing a master's degree, go and do your master's study over there because there's only 5 herbarium specimens from it in the entire state of California, plus the various other entities that are involved in the consortium. Very, very powerful tool to focus our energies, focus our abilities, and our financial abilities, really, in a very precise way to get the most done. Likewise, niche modeling approaches can be used to help us know where to look for plants. We do a lot of work in Southern California assessing allegedly rare plants. Some of them turn out to not be rare, including at least one that was thought to be extinct, and we now have found 5 or 6 or 7 populations of it, and it's actually doing quite well using a niche modeling approach. identifying places in the complicated topography that is California, really, to identify where to go to look, even to what soil types to look on. Kay mentioned seed banking quite a bit, and seed banks are terrific. When I do tours of our seed bank facility, I tell people that they're in a small room with hundreds of millions of individuals. Luckily, almost all of them very small and very dormant. And people seem to really enjoy that kind of metaphor. I think we can readily agree that when we make a seed collection that is scientifically valid and responsible, that is taking account of genetic variability and not over-collecting, that we have likely have a much better sample of genetic diversity of plants than we can, in almost all cases, ever have in our ex situ living collection. And of course, seed banks have a huge number of uses and will have even more uses going forward, is my prediction. We use our seed bank to grow plants for display in our garden. Obviously, restoration, which Kay said quite a bit about. I want to mention a really exciting and very important aspect of seed bank use, which is for documenting evolutionary responses to climate change. So we definitely have seeds in seed banks that were collected tens of years ago, 50 years ago, et cetera, longer ago. And for short-lived species, we may expect that over 50, 60, 70 years, over the space of decades, instead of hundreds or thousands of years, we may see evolutionary change happen in response to climate change. So the idea that we can do experimental outgrows of seeds of different ages and look at how their traits have changed over this time is really very exciting way to think about it. I think that seed banks have very important roles to play in restoration and in our what are going to be very complicated responses to climate change. And there's also the possibility that seed banks will provide for species resurrection. We have in our herbarium a couple of cubby holes that we call the dog and pony show, which are specimens that we keep handy to show to people who come to visit. And a couple of the things that we have there are specimens of plants that, as far as anyone knows, are extinct and are very likely to be extinct. Not just absence of knowledge, but near knowledge of absence. And those are very poignant examples. These are plants that lived, that demonstrated a unique way to be a living organism on planet Earth and are now not with us anymore to share that knowledge. If only we had seed banked those species, we would be able to contemplate resurrecting them, and we will be able to do that going forward, as I'm afraid that some of the rare plants in our seed banks will not meet a positive fate in nature. Again, I want to say that all of what I've said involves networks, collaborations, groups like CPC and Botanic Gardens Conservation International working together. No one garden can do it alone, but we all must be agents of change. I want to cycle back and say that the conquering green blindness I see as absolutely essential. We must embrace that, the challenge of making the knowledge of how important plants are, how critical they are, shared by our citizens, our fellow citizens who don't automatically get it like many of us in this room. Thank you very much.
Thank you, Dr. Havens and Dr. Mcdade. We have about 6 minutes for questions for this panel, so if you have questions for the three of us, we'd be happy to entertain those.
I'm curious as to whether or not there is ever a point where you have to accept that investigating and trying to propagate a species is beyond saving?
I haven't encountered it yet. Oh, sorry. I haven't encountered it yet. I've worked with a lot of basket cases, and we've always been able to do something. There may be some species that we just can't save, but I think the vast majority of them, we have the ability with the proper funding to do so.
I would say that in many cases, we really have advantages with working with plants compared to animals in terms of the question, the very excellent question that you raise. Many plants are hermaphrodites. and are not like vertebrate animals that have problems reproducing by themselves. Of course, there are plants that are dioecious that have the sexes on different individuals, and those may well present similar challenges for things that are extremely rare. Yeah.
I'm wondering how for Dr. McDade, how the current drought conditions in California are affecting your thinking about the importance of your work.
Well, thank you for that question. It absolutely highlights the importance of focusing on sustainable landscapes in Southern California and California in general. It has one of our missions is to increase the public appreciation of California native plants and also to encourage their use in home and commercial landscapes. And the fact that people's water bills are going up even as their lawns are still turning brown has a way of getting people's attention in terms of thinking about making those conversions. At the same time, it is extremely challenging to run a 86-acre botanic garden, and we do grow plants from other parts of California that benefit from a bit of summer water every now and then. So we've had some real challenges maintaining the garden, to be entirely candid. It's a very challenging environment in all of California right now, although it's still amazing how many people seem to be in denial that water doesn't just come out of their pipe regardless of how long they leave it running or how much they're willing to pay for it. It's a very interesting time. I think that part of plant blindness or an extension of plant blindness is also natural resource blindness. I think people have a hard time understanding that some resources, not the sun, but some resources really are extremely limited and require shepherding.
I think we have time for one more question.
Is there such a thing as a bad plant or a terrorist plant where with an invasive species or an incredibly aggressive plant, it is the growth of one plant impedes the diversity that you're trying to protect?
Well, California is the poster child for invasive plants. Many plants from other parts of the world do extremely well in Southern California. And that's true also of some parts of the desert Southwest in general. And it is partly the competition problem that you allude to, where we have found that the invasive grasses in our part of the world tend to germinate and get established and form a solid thatch over the ground before native annuals can get going in the spring, because they simply have slightly different strategies in terms of timing of getting going. That's a major problem. But there also are major problems associated with these native invasive plants, especially grasses, altering things like the fire regime, because they tend to lay down. It's like strewing completely dry hay over a slope, say, and then setting it on fire. The presence of all of that extra biomass, all of that dried down biomass, really increases the fire intensity and the fire frequency as well. So it's really an extremely major issue in a lot of parts of California and in the Southwest.
Yeah, and if I can take 1 second to follow up, we have a program working with many others called Native Winners, where we're searching for native plants that persist in highly invaded sites, and these might be particular species or even ecotypes of species, and we're propagating those that do well in competition trials with cheatgrass and some of the other weeds that Lucinda was mentioning, and promoting those for restoration use.
I think we're out of time, but I want to thank the panelists again, and thank you everyone for your attention.